Mr. Chairperson, sir, I'm honored to be invited to deliver this guest lecture on hepatitis E recent developments. Hepatitis E is now recognized as a global health problem and no more a disease of those third world countries. In this talk, I shall focus on the long way we have traveled to reach what I call today the Hepatitis E 2010. This talk is in 3 short segments to remind this august gathering of where from we started, where we have reached today, and where we have to reach finally. I shall take you back to some stories and images of late 80s which had never been heard or seen by the scientific community before. I feel nostalgic to go down my memory lane of these 30 years since I recognized the first ever epidemic known in non-B hepatitis in November 1978, just 3 months after I left PGI Chandigarh to set up a clinical unit of gastroenterology in Kashmir Valley. Right from the onset, I was more than convinced that we were dealing with a new disease, and that's the reason why I incorporated the statement, possibility of another human hepatitis virus distinct from post-transfusion non-A, non-B type, in the title of the award-winning plenary session paper presented at the 20th Indian Society of Gastroenterology meeting at Pune on October 12, 1979, and soon published with the same title in the American Journal of Medicine in January 1980. While this news was received with great interest by Indian and most interested scientific community, it was not without skepticism from some international experts, and I shipped thousands of sera to many interested laboratories only to silence the skeptics. Many young physicians have asked me as to how I could conceive the existence of a new disease based on these data. 3 aspects of 1978 epidemic Kashmir needs special comment. First, sheer magnitude of problem was unprecedented. I remember no less than a mini tsunami-like state of affairs on my first visit to the region of the epidemic. The disease had caused colossal human suffering and loss of life, and an estimated 52,000 patients had icteric disease with around 1,700 deaths. Was one of the largest epidemics of hepatitis recorded for all times at that time. Second, we conducted an intensive house-to-house survey to meticulously document evolution and progression of the epidemic by registering every new case of hepatitis. This type of survey was unprecedented in history and was made possible by the service of over 500 healthcare workers. I must mention that the region involved by the disease had extreme weather conditions and most primitive health care facilities, and it was extremely hard to do field studies under this trying situation. Also, the laboratory facilities were non-existent, and we had to get an outreach help for advanced lab testing. I funded this project to a substantial proportion from my private clinic income. Third, the epidemic had many unique clinical and epidemiological features which could not be explained by any known agent, including hepatitis A. One of the most distinctive features of hepatitis E was highly compressed epidemic curve, lasting for around 7 weeks, suggesting a single common source mode of spread. There was convincing evidence that Kashmir Epidemic 1978 was related to consumption of fecally contaminated drinking water. In fact, drinking water sources of this region had been very busy sites where public latrine sewage flows, garbage of whole locality was dumped, utensils and linen was washed, children swim, and locals buy fish as seen in the boat in the background. Following epidemics of hepatitis C, secondary waves of hepatitis usually did not occur, suggesting that person-to-person contact transmission was not a major factor in the evolution of the outbreaks. These and further data about person-to-person transmission of hepatitis E virus infection have raised major controversy about how disease spreads in developing countries in non-epidemic setting, and this issue is yet unresolved. One of the most perplexing features of Kashmir Epidemic 1978 was age distribution of cases of viral hepatitis. Majority of icteric cases occurred in the age group 11 to 40 years, leaving children and elderly relatively unscathed. These data were in sharp contrast to hepatitis A, where most infections occurred in the first decade of life. This pediatric paradox was a unique clinical feature of hepatitis E and remains unexplained as of today. Second unique phenomenon of hepatitis E was increased incidence and severity of disease in pregnancy. Incidence of disease was 8 times higher in pregnant women than in the 2 control groups. The incidence of disease was higher in all 3 trimesters to that in non-pregnant women. Fulminant hepatic failure occurred over 13 times more in pregnant women than age-matched control. It occurred in astonishingly 44.4% in third trimester pregnant women. In fact, increased incidence of severity of disease in pregnant women during epidemics and sporadic infections of hepatitis E are the most challenging problem for healthcare delivery in the developing countries. Further progress of hepatitis C research was an example of extreme zest for human knowledge and self-experimentation. From August to September 1981, non-A, non-B hepatitis outbreak occurred in Swat military camp located in Afghanistan. A team from the Institute of Poliomyelitis, Moscow, led by Dr. Balian, investigated this epidemic. The investigators were impressed with the fact that this epidemic closely resembled the Kashmir Epidemic 1978. To determine the causative agent of this outbreak, Dr. Balian himself ingested pooled stool extracts from 9 patients. He developed typical clinical signs and symptoms of acute non-A, non-B hepatitis accompanied by enzyme abnormalities 36 days post-inoculation. 27 to 30 nanometer spherical virus-like particles were visualized in stool samples collected on days 28, 43, 44, and 45 after inoculation. This set a stage for the search of an agent and its further characterization. Excellent data on element transmission biophysical properties of the pathogen were published from CDC during the period 1983 to 1999. However, the pathogen frustrated molecular biologists as recoveries of virus-like particles from stool specimens were insufficient to clone and sequence the virus. We were advised to dump large specimens of stool samples from patients into our deep freezers, and many of us, including me, did it faithfully at the great wrath of the nurses and the laboratory staff. A breakthrough in this area occurred with the observation that while virus-like particles are rare in stools, the bile is loaded with infectious material. This observation led to the cloning and sequencing of the agent, and a full-length genome was subsequently sequenced and a serological test developed. Improved antigen expression made the serological test valid for clinical and epidemiological purposes. We have come a long way as hepatitis E is as of 2010. Hepatitis E is a small, non-enveloped, single-stranded RNA virus and is the sole member of the genus Hepivirus in the family Hepiviridae. 2 major species of the virus are recognized: avian hepatitis E virus and mammalian hepatitis E virus. Avian hepatitis E virus causes big liver and spleen disease in chicken, but no human disease. Mammalian hepatitis E virus causes disease in human beings. Hepatitis E virus has considerable genomic diversity, and 4 major genotypes of the virus have been identified. Genotype 1 and 2 are limited to human host and is the cause of epidemic and endemic human infections in developing countries. No animal reservoir has been identified for these genotypes. Genotype 3 and 4 are ubiquitous in swine and wide spectrum of animal kingdom, and has entered human population and caused human disease through ingenious mechanism of foodborne zoonotic transmission. Hepatitis C is an important public health concern and is a major cause of enterically transmitted hepatitis world over. Based on seroprevalence data, an estimated 1/3 of the world population has been infected with hepatitis E. Genotype 1 and 2 cause 3 epidemiological patterns of disease. Hyperendemic zone is riddled with repeated large waterborne epidemics, and over 50% of endemic acute hepatitis in these regions is related to hepatitis C. The disease involves tens of thousands of cases and is the cause of considerable morbidity and mortality, posing a major public health problem in hyperendemic regions. Indian subcontinent falls in the hyperendemic zone, and an estimated 2.2 million cases per year of hepatitis C occur in India alone. Endemic zones have not been recorded any epidemics of disease, but around 25% of endemic acute hepatitis in these regions is caused by hepatitis E. Egypt, for some reasons, has distinctive pattern of disease exactly resembling hepatitis A, and most infections in this country occur in children. Hepatitis C genotype 3 and 4 infections are now recognized as significant clinical problem in industrialized countries. Genotype 3 causes sporadic human infections in UK, many European countries, United States, Australia, and few South American countries. Genotype 4 causes human infections in most parts of China and many South Asian countries. Both genotype 3 and 4 are endemic in Japan and cause human infections. Hepatitis C in developing countries is nearly always caused by genotype 1 and 2, and this slide depicts the modes of spread of hepatitis C in these countries. Gross water contamination is the major source of repeat epidemics in hyperendemic zones. Hepatitis E viremia in otherwise asymptomatic persons is not an uncommon occurrence. Viremia can be protected in a small percentage of patients, and substantial percentage of patients may have subclinical infections. Post-transmission hepatitis C has been documented by our group. Hepatitis C virus has a distinction of universal vertical transmission to the fetus and newborn when mothers are infected in second and third trimester. This vertical transmission causes high fetal loss, and most newborns develop hypoglycemia and hypothermia as a manifestation of massive hepatic necrosis. We have preliminary data that severe fetal disease may be related to increased severity of disease in the mother, which may be another example of mirror disease of pregnancy. There is no consensus about the person-to-person transmission of hepatitis C in endemic disease in developing countries, and we need further data to sort out this issue. Swine in India are infected with hepatitis C genotype 4. However, this infection has not been documented to occur in humans in this country. Hepatitis C in industrialized countries is peculiar zoonosis adapted to humans. Indigenous hepatitis C is now recognized as a clinical problem in industrialized countries. Several animal species, especially domestic swine, wild boar, and wild deer, are found to be reservoirs of genotype 3 and 4 in these countries. Human infections occur through intake of uncooked or undercooked meat of the infected animals and pig livers and sausages made from these livers and sold in the supermarkets. Approximately 2% of the pig liver sold in local grocery stores in Japan and 11% in the United States test positive for infectious HIV RNA. Recent data have also identified few cases of transmitted hepatitis E in Europe and Japan. How common is this phenomenon needs further studies. We know so much about hepatitis C in our community. The disease occurs in young adults and shows peculiar relationship with pregnancy. Hepatitis C superinfection in patients with chronic liver disease often causes decompensation of stable chronic liver disease and high mortality. Indigenous hepatitis C in developing countries is often misdiagnosed as drug-induced liver disease. This has been due to a lack of awareness of hepatitis C among clinicians in these countries. Most such infections have been reported in middle-aged and elderly people. Disease is generally more severe than reported in developing countries, and 15% of such patients develop hepatic or non-hepatic manifestations. Fulminant hepatic failure develops 8 to 11% of hepatitis C in these countries. Superinfection in patients with underlying stable chronic liver disease often presents with hepatic decompensation and mortality approaching 70%. Phenomena of increased severity in pregnant women due to hepatitis C has not been described in these countries. Unlike hepatitis B and C viruses, hepatitis C virus is generally believed to cause only self-limiting acute viral hepatitis that does not result in chronicity. However, recent studies reveal that hepatitis E genotype 3 can cause chronic hepatitis in immunosuppressed organ transplant recipients. A French group studied 14 cases of acute hepatitis C virus infection genotype 3 in patients receiving organ transplants. A developed chronic hepatitis C as evidenced by persistently elevated aminotransferase levels, detection of hepatitis E virus RNA in sera, and microscopic evidence of chronic hepatitis. A number of reports have identified chronic hepatitis C in liver transplant patients, which have culminated in graft failure needing retransplantation. Persistent hepatitis C virus infection was also identified in patients infected with hepatitis C via blood transfusion during chemotherapy against T-cell lymphoma. Reactivation of hepatitis C virus infection was also reported in patients with acute lymphoblastic leukemia 14 weeks after allogeneic stem cell transplantation. Taken together, these recent studies indicate that hepatitis E genotype 3 infections are often cause chronic hepatitis in immunosuppressed patients, and this can rapidly progress to cirrhosis and end-stage liver disease. Recent reports of antiviral therapy in chronic hepatitis C have been most exciting and may open a possible way for us to manage severe acute hepatitis E superinfections and severe hepatitis C, especially in pregnant women. Pegylated interferon alpha-2 12 weeks therapy causes rapid and sustained virologic response in liver transplant patients and those with immunosuppressive diseases. Chronic hepatitis C in patients with renal transplant in whom interferons are contraindicated, show a similar sustained biological response with oral ribavirin therapy in most instances. The N-terminal truncated capsid protein containing amino acid residue 111 to 660 can self-assemble into empty virus-like particles when expressed in baculovirus. A major breakthrough in hepatitis C virus research is the recent determination of 3D crystal structure of virus-like particles. These 3D structure studies reveal that hepatitis E capsid is formed by capsomeres consisting of homodimers of a single capsid protein forming the virus shell. The homodimers protrude from the virion, interact with the host cell, and bind to the cellular receptors to initiate virus infection. The 3D structure of hepatitis C virus will undoubtedly advance our understanding of the mechanisms of hepatitis C virus assembly and entry, and it will also aid in ongoing efforts for the development of an effective vaccine and antivirals against hepatitis E. The lack of cell culture for the hepatitis C virus hinders initial vaccine development. However, recombinant DNA technology has been used to produce ORF2 encoded capsid proteins in insect cells. 2 candidate vaccines have successfully completed phase 2 trials and ready for prime time. SmithKline-Beecham licensed baculovirus-expressed ORF2 proteins from a Pakistani strain of hepatitis C virus. A randomized trial was conducted in Royal Nepal Army, found the vaccine to be safe and efficacious in the prevention of clinical disease. Another large randomized trial of vaccine prepared from recombinant proteins from HEV-ORF2 viral capsid expressed in Escherichia coli. The vaccine named as HEV-239 has recently been completed in China. Vaccine efficacy after 3 doses was 100%. The vaccine was well tolerated, effective in the prevention of hepatitis C in general population in China. These vaccines could prove very useful in the prevention of morbidity and mortality from hepatitis C virus infections among pregnant women and patients with chronic liver disease in endemic areas, and those organ transplant patients or immunosuppressed patients who may contract hepatitis C virus infection genotype 3 in industrialized countries. Mr. Chairperson's son, we have come a long way about hepatitis C story over the last 30 years since the first description of the Kashmir Epidemic 78. However, a number of important issues remain unanswered. These can broadly be divided into 4 categories, namely introduction of improved assays to detect hepatitis C virus infections, better understanding and answering of many perplexing issues about the epidemiology of hepatitis E, develop treatment strategies of hepatitis C virus infections and management plans for those with severe infections, especially liver failure, and development and implementation of effective preventive strategies, which is especially hepatitis C vaccine. As we proceed to fulfill the challenges imposed by the hepatitis C virus, a public health problem of global importance shall eventually be conquered. Thank you very much for your patient hearing.